Number 1. Brain data collection. Capturing the brain in action. Today, we begin with the first stage of computational neuroscience, brain data collection. Before we can analyze the brain using artificial intelligence, AI, or machine learning, ML, we first need high-quality brain data. Think of this as building a house. The data are the foundation. If the foundation is weak, the entire analysis will be unreliable. What do we measure? Before selecting an instrument, we must decide what type of brain information we want to measure. Different techniques measure different aspects of brain function. Electrical activity. Neurons communicate through tiny electrical signals, measured by EEG and ECOG. These techniques tell us when brain activity occurs with excellent precision. Magnetic activity. Whenever electrical current flows through neurons, it also produces tiny magnetic fields, measured by MEMI. MEG records these magnetic fields to study brain function with excellent temporal accuracy. Hemodynamic activity. Active brain regions require more oxygen and nutrients. As a result, blood flow increases to those regions, measured by fMRI and fNIRS. These methods measure changes in blood oxygen rather than electrical signals. Metabolic activity. Brain cells consume glucose and oxygen to produce energy, measured by PET. PET measures brain metabolism and is widely used to detect neurological diseases. Behavioral and cognitive. Brain activity is often compared with a person's performance during a task. Examples include reaction time, accuracy, choices, errors. These measurements help explain how brain activity influences behavior. Physiological signals. The body often reacts together with the brain. Researchers also measure heart rate, skin conductance, respiration, pupil size, muscle activity. These physiological signals provide additional information about attention, stress, emotion, and cognitive workload. B. Major brain data collection methods. 1. EEG, electroencephalography. EEG records electrical activity from the scalp using electrodes. It provides excellent temporal resolution, milliseconds, moderate spatial resolution, portable and relatively inexpensive equipment. EEG is widely used for epilepsy research, sleep studies, cognitive neuroscience, and brain-computer interfaces, BCI-2. MEG, magnetoencephalography. MEG measures the magnetic fields generated by active neurons. Its advantages include excellent temporal resolution, good spatial resolution. However, it requires highly sensitive squid sensors and a magnetically shielded room, making it one of the most expensive brain imaging techniques. 3. fMRI, functional magnetic resonance imaging. fMRI measures changes in blood oxygen levels, known as the BOLD, blood oxygen level dependent signal. Its characteristics include good spatial resolution, millimeter level, moderate temporal resolution, seconds. Completely non-invasive fMRI is widely used to identify where brain activity occurs during cognitive tasks. Number 4, MRI, structural magnetic resonance imaging. MRI provides detailed images of brain anatomy. It offers excellent spatial resolution, high-quality structural images. MRI is commonly used for structural brain mapping and EEG source localization. 5. PET, positron emission tomography. PET measures brain metabolism using radioactive tracers. Its characteristics include good spatial resolution, low temporal resolution, minutes, involves exposure to radiation. PET is frequently used in clinical neuroscience to study Alzheimer's disease, Parkinson's disease, and brain tumors. Number 6, fNIRS, functional near-infrared spectroscopy. fNIRS uses near-infrared light to measure changes in blood oxygenation. Advantages include portable, affordable, suitable for bedside and infant studies. Its limitation is that it mainly measures activity from the outer surface, cortex, of the brain. Number 7, ECOG, electrocorticography. ECOG records electrical activity directly from the surface of the brain. It provides very high spatial resolution, excellent signal quality. Because electrodes are placed directly on the cortex, ECOG is an invasive technique used mainly during neurosurgery. Number 8, eye tracking. Eye tracking records where a participant is looking. It measures eye movements, fixations, saccades. Eye tracking is widely used to study attention, perception, reading, and decision-making. 9. Behavioral data. Behavioral data describe how participants perform during experiments. Typical measurements include reaction time, accuracy, choices or responses, subjective ratings, error types. These measurements are essential for linking brain activity to human behavior. Now, number 10, physiological sensors. Physiological sensors record body responses that accompany brain activity. Common measurements include heart rate, ECG, skin conductance, GSR, respiration, pupil size, EMG, muscle activity. These signals provide valuable information about stress, fatigue, emotional state, and mental workload. C. Typical experiment setup. A typical neuroscience experiment follows a standard procedure. The participant wears an EEG cap, sits comfortably in front of a computer, and responds using a response device. During each trial, a stimulus is presented. Brain activity is recorded. Behavioral responses are collected. All devices record data simultaneously. This setup ensures accurate and reliable data collection. D. Data synchronization. In modern neuroscience experiments, multiple devices record data at the same time. These may include EEG, fMRI, eye tracking, response device. All devices are synchronized using the same clock. The moment when the stimulus appears is marked as time, t equals 0. The timeline usually extends from -1 to +3 seconds, allowing researchers to compare signals recorded by different instruments with precise timing. E. Data output and formats. Different instruments save data using different file formats. Examples include EEG MEG IDF.FIF BDF, MRI fMRI NI.GZ DCM, PET DCM, behavioral data.CSV TSV XLSX, eye tracking DF ASC HDF5, physiological data.CSV TXT, event markers.TSV TXT. These files are later analyzed using software such as MATLAB, Python, EEGLab, Brainstorm, SPM, and Fieldtrip. Number F, key considerations. High-quality neuroscience research depends on careful planning and good experimental practice. Researchers must ensure ethics and consent, obtain informed consent, protect participant privacy, and maintain data security. Participant safety and comfort, minimize discomfort and ensure participant well-being. Task design, use reliable, valid, and well-controlled experimental tasks. Calibration, properly calibrate and test all equipment before recording. Quality control, check recordings for noise, artifacts, and missing data. Data storage, store data securely, maintain backups, and organize files properly. Metadata, record complete experimental details, including participant information, equipment settings, and task parameters. NAHRC summary, brain data collection is the foundation of neuroscience research. It involves measuring electrical, magnetic, hemodynamic, metabolic, behavioral, and physiological signals using specialized techniques such as EEG, MEG, MRI, fMRI, PET, fNIRS, ECOG, eye tracking, and physiological sensors. Accurate data collection, precise synchronization, proper storage, and rigorous quality control ensure that the data can be confidently used for signal processing, brain mapping, machine learning, clinical diagnosis, and brain-computer interface applications.